We are lucky to have a presentation from Jehoshua Sharma, who is a PhD student from Trinidad and Tobago, who is currently studying the fungal pathogen Candida albicans at the University of Guelph. His current research looks at compounds that can enhance the activity of known drugs or that can disarm the pathogen of virulence mechanisms that make them harmful to the human host. He is also developing novel CRISPR variants to study the functional genomics of a drug-resistant Candida albicans isolate. Outside of the lab, Joshua is a co-founder of cGEM, a Canadian-wide initiative connecting research groups tasked with using synthetic biology to solve real-world problems. His aim is to give students access to the tools they need to conduct research independently and communicate the research on a wider scale to the public. Now, unfortunately, Joshua wasn't able to join us today as planned, but luckily he provided us with a recording so that we still get to learn from him. So with that, we'll learn from Joshua about pathogens and how fungi can be pathogens.
Hi, everyone. My name is Jehoshua Sharma. I'm a PhD student at the University of Guelph, and I'm here today at our CIS Sciences, the Fungi, Good, Bad, and the Ugly event, to talk to you about fungal pathogens and really taking a look at the more microbial and bad nature of fungi. And even though I'm not with you here in person today, I'm super excited that I still got a chance to send in a video with this presentation. And if you have any questions or comments that may follow, feel free to send me an email, follow me on Twitter. I'll be here and available to take and answer questions. Now, let's get started. Historically, fungi tend to really be thought of as an agricultural threat. We had farming conditions whereby, we have one species like wheat or rice that are planted for acres, and there was and continues to be a significant vulnerability with that. When one plant gets infected by a pathogen, it quickly spreads across the field, and it results in endangering the entire harvest. Now, we've learned these lessons in instances where the fungal like Oomycete, Phytophthora infestans actually decimated potato crops in the 1840s, leading to what we call the Great Irish Potato Famine. These lessons we really continue to learn today when we deal with fungi that can infect and destroy important crops like wheat and rice. We have fungi like Magnaporthe and Fusarium species that can actually infect these crops. And once inside of the plant, they secrete toxins that kill the surrounding tissue. It's these fungal outbreaks of food crops that result in the loss of their viability. And it really reduces our global capacity to feed people by 8.5%. That means that there's approximately 650 million people that go hungry each year due to fungal pathogens. And now moving on to something that you might not be as aware of is that unlike plant pathogens, human fungal infections had been really rare in the past. It wasn't really until the 1950s when antibiotics, immunosuppressive agents that all revolutionized modern medicine created these conditions where large amounts of bacteria were just indiscriminately killed off in the humans. And it left fungi with new niches to colonize. And so whilst we have medical advances that started to save countless lives, they also enabled this beginning of a shift in the epidemiological landscape of the type of microbial infections that actually pose a threat to us. Now, on your right, you'll see this pie chart that highlights fungal infections and death rates in 2016. And while you might think, oh, mortality looks low, at 0.15%, this diagram is actually a bit misleading, as over 1 billion people are afflicted with fungal pathogens globally. And so 1.5% of 1 billion leaves 1.5 million lives that are actually lost each year due to these fungal pathogenic infections. And really compare this to the 1.8 million lives that were lost to COVID-19 in 2020. I hope that with this kind of description, you can start to see the cause for concern. Now, I really don't have time to go into all of the human fungal pathogens, but there are a few major ones, all that are associated with high mortality rates if the infection becomes severe, that I want to bring up and tell you about. The first set are Aspergillus species. These are actually really common airborne fungi that once they enter your lungs and they're able to overcome your immune cells, become responsible for about 3 million cases of chronic lung disease. And they're also a substantial cause of fungal-associated asthma, which affects 10 million people worldwide. Next, we have Candida species. Now, Candida species tend to be human symbionts. This means that they're microbes that naturally live inside of all of us. However, despite this fact, it's a part of the healthy human body, yes, but it causes infections in our mouths or vaginal regions due to its overgrowths. And also they become really problematic when medical treatments create these environments where Candida albicans proliferates unchecked. We have either the treatment with antibiotics, again, killing off a lot of bacteria, or we have the insertion of catheters where they can grow and form biofilms. And it's these types of infections that lead to death due to the development of intense bloodstream infections. And these bloodstream infections carry the microbe around the body and they cause invasive candidiasis in infected organs. And honestly, it's associated with mortality rates as high as 70% in some countries. With these two, I'm hoping you're starting to see the bad with fungi. Now, the last fungal pathogen that I want to talk about are actually the Cryptococcus species. These microbes that are commonly found in the soil can cause very serious infections that begins with the inhalation of desiccated yeast cells or spores from the environment. And once they've established an infection site in your lungs, they can be disseminated to the central nervous system. This will lead to fungal meningitis as well as bloodstream infections. And what's really interesting to note here is that Cryptococcus species tend to kill a bit more than tuberculosis does each year. Yet you hear and far more about tuberculosis and our push to eradicate it than you often hear about fungal pathogens. And what I'm hoping to do here is not really scare you into thinking, oh my goodness, I'm going to die from a fungal infection. But I want to highlight, that fungi in general are it's these groups of organisms that cause very serious diseases, and they're so often neglected. And this is something that all humanity should really be paying more attention to. Now, outside of fungal infections, we also have many animals that face diseases that seem almost apocalyptic to us. And fungal pathogens are really diverse in the ways in which they go about being a pathogen. One of the most common examples is the infection of ants by the Ophidiocordyceps fungi. Now, this is an infection that really begins when a fungal spore attaches to and degrades an ant's exoskeleton. Then the fungi begins growing in a yeast state in the bloodstream, and eventually it grows to such a level where it can grow between the ant's muscle cells and then begin to form this dense network of fungal cells that just completely hijack the ant's body. Now, this is some really scary, ugly stuff, but with the ant under the fungus's control, it climbs up off the ground and then really clamps on with its mandible to just like a vein on a huge leaf. And as you can see from the pictures taken, the fungi begins to grow this fruiting body that just like protrudes out of the base of like the ant's or like the tarantula's neck. And it's the height of like the insect that allows for the fungal spores to be carried on the wind and onto like their next target to repeat the process just all over again. Now, a little less horrifying, but still extremely concerning, is the infections of amphibians by the Batrachochiretrium, that's a word, fungus. Now, this is a skin infection that leads to the thickening of the amphibian skin, which ends up disrupting the osmotic balance in its body, which is honestly, as you'd imagine, never good. It leads for these amphibians, unfortunately, huge series of cardiac arrests and deaths, affecting animals from frogs to salamanders. And although the disease is less horrifying than the Ophidiocordyceps that I just described, this fungus is actually linked to the increasing decline of amphibian populations globally. And it's currently thought that with this increase in infections and death that's caused by climate change, with these increasing global temperatures, these amphibians just end up being more susceptible to these infections, and the geographical range of the fungus is starting to grow. Now, as alarming as all of these examples are, we have some tools in our back pocket that really allow us to treat a lot of these infections for both plants, insects, and animals. Most of us have heard about antimicrobials, which are compounds that really limit microbes by stopping their growth or killing the cells directly. With fungi, we have antimicrobial compounds called antifungals. And antifungals are amazing because, well, they really help us treat these pathogenic fungal infections I've been describing to you. And what I'm going to do is just walk you through some of the more common classes of antifungals using a handy dandy fungal cell cartoon. So There are three main classes of antifungals that I want to talk about, and they're all represented by the red squares in this figure. First, we have the azoles. They're antifungals that prevent fungal growth by inhibiting their ability to create new components that are essential for the fungal cell membrane. Without it, the fungal cell can't really continue to grow, and so it just stops fungal growth. Then we have the paulenes. The paulenes are antifungals that really just physically bind to components of the cell membrane. It forms these antifungal-induced pores in the membrane and causes the cell to literally rupture, killing it. And finally, we have the echinocandins. These are a cool group of antifungals that, similar to azoles, they prevent the creation of new cell components. But instead of targeting the cell membrane, they target the cell wall. And without a cell wall, it essentially really weakens or removes the pathogen's ability to survive in its environment. And the echinocandins end up killing the fungal cell. Now, I really wish that this victory was the end of knowing how we deal with fungal pathogens, but unfortunately it's not. And there's something else I would like you to remember, which is that sometimes fungal pathogens can get random changes in their DNA through the process of evolution and natural selection that allows them to really, resist the antifungal's lethal effects. And it gives us a new problem where we know it as antifungal resistance. Now, often fungal pathogens have a highly plastic genome that allows them to generate a huge amount of genetic diversity, helping encourage this resistance. And these resistant fungal pathogens tend to have a lot of mutations that let them resist the traditional drug treatment options. The mutation usually results in either altering the drug target, so they can change it and the drug ends up not being able to have any activity. They can upregulate the expression of the drug target itself. And when you create more targets in the cell, you need essentially a higher concentration of antifungals to achieve the same effect. And lastly, they can just simply export, the drugs out of the cell via efflux pumps. And if there's no antifungal drug in the cell, then there'll be no antifungal effects to be had on any of the pathogens. And so this actually also isn't where our problems stop, because not only do we have issues dealing with the fungal pathogens that we know, but a lot of human activity is actually causing the rise of new and emerging fungal pathogens. And, everyone here knows that we have an incredible amount of data showing how human activity has led to the increased warming of our planet. This creates a huge set of problems, but specifically for this presentation, it impacts fungal pathogens in a really interesting way. This is simply because the ability of most fungi to infect humans is thought to be as a consequence of the way they're adapted to a warmer ecological niche. We often say that fungi tend to be accidental pathogens rather than intentional ones. And the key factor that provides a barrier to infection for warm-blooded mammals specifically is our core temperature at 37 degrees, because most fungi are just simply unable to grow at that temperature. This is known as thermal restriction, and it's really thought to be our greatest weapon against. I think you can see where I'm going here with this, but unfortunately, with the warming of the planet, fungi now also have to adapt to warmer temperatures, and it's this that increases these issues between us, because once they've adapted, they can now infect us with relative ease. Now, for example, in 2009. There was a new species of Candida pathogens called Candida auris, you're seeing in the yellow on the screen. And it was identified globally and thought to originate from the environment. Candida auris, naturally possesses genes that make it pathogenic to mammals. But because of the thermal restriction that used to exist, it never really was a problem for us. And it's currently thought that the increase of global temperatures is what allowed it to be able to now only start infecting us. Now, additionally, remember earlier when I said that agriculture causes some problems for us in the way that we have it set up? Well, one of the ways we ensure that fungal pathogens don't actually destroy all of our crops is the widespread use of antifungals. Now, the widespread use of antifungals do protect a lot of our food sources. And if you'd recall, on my slide that I said just earlier, when it comes to antifungal use, it also leads to antifungal resistance. And so unfortunately, something that helps us also hurts us in another way, because this process of spraying all of our crops with these antifungals to protect them leads to really, really high rates of environmental antifungal resistance. And so something else to know is this Candida auris species I was just telling you about. it's also highly resistant to most forms of the antifungals that we have. And so whilst it was only recently discovered, it's also a pathogen that has a really high mortality rate. And, there are cases like this that are starting to pop up and become more common. And we have another example where Aspergillus species that I was telling you about earlier, now people are being found with antifungal resistant Aspergillus species in their body, whilst they've never been exposed to antifungals before. And it's thought that it's this use of antifungals in our environment that contributes to this growing problem. And so please, you should expect to hear more about how emerging fungal pathogens are really coming about and really becoming problematic due to the human specific impact that we have on the environment around us. Now, that's definitely not a note that I'm going to end on, because it wasn't bad or ugly, but straight up menacing. Where I really want to end off is what current scientists like myself are doing to address the threat of fungal pathogens, both old ones and the emerging ones. And it's a bit different than what I've been talking about so far, because it has to do a lot more with the genetics and the physiology of the pathogens. So we do all of this by following something called the central dogma, where molecular biologists believe that DNA makes RNA, which then goes on to make protein. And we can infer the interactions between these three based on the output of the cell that we're studying when one of them are disrupted. In my lab, we do a lot of genetic manipulation at the DNA level using something called CRISPR-Cas9. And in short, Cas9 is a protein that can be guided to precisely target specific DNA sequences, and it mediates these double-stranded breaks in the DNA. We then are able to use CRISPR to add genes that may be of benefit, but we mostly use it to really just knock out genes and study the resulting virulence, pathogenesis, drug resistance, or even really the basic biology of the organism. And once we end up mutating a pathogen how we'd like it, we can then put it in a mouse, so we can put it in a test tube with drugs, and we can record how well it functions, even though we've altered the protein in some way. And in its entirety, we have different ways to study these mutants, and we can look at the genetics, proteins, and even overall metabolism. This sort of combined approach allows us to identify everything from potential biomarkers, and it even allows us to describe the fine-tuned interactions that these microbes would have in regular infection settings. And where we stand right now is amazing, because we can quickly analyze any organism, again, old or new. We can find out what are its potential disease phenotypes, what toxins or hormones that make it pathogenic, or even, what parts of the cell are really essential for its specific survival. And once we understand those things, we can then create new compounds that end up making very good drug targets. And this is the way that you should really be thinking about how people study fungal pathogens. And it's really exciting, again, because it results in us having rapid, specific targeting of these pathogens, and it allows us to effectively treat a lot of the hosts, both human and non-human, with limited detrimental effects to them. So honestly, with that, I just want to say thank you so much for your time and attention today. And if you have any questions, feel free to find a way to reach out to me. Thank you, RCIS, for the opportunity, and I will hopefully see all of you soon. Bye.
So that was such an amazing presentation from Joshua. And as a reminder, if you have any questions specifically for Joshua, that he provided his contact information, so feel free to reach out. So thank you, Joshua, again for that.